Now it's all right? Am I audible? Yeah. So all well? Doing good? Okay, ji. Let's start the today lecture. Okay? Have a look. So we are starting with mitochondria. Right. Okay. One student asked difference between plasma membrane and plasmodesmatite. Message, na? You told? You just messaged me or someone else? One message came to my mobile. I just answer also. Okay. So if we talk about mitochondria, right, Ji? So mitochondria is also called powerhouse of the cell. Mitochondria is called what basically? Powerhouse of the cell. And if you talk about mitochondria, along with, or you can say simply, it is a semi-autonomous cell organelle. It is a semi-autonomous cell organelle. Why it is a semi-autonomous cell organelle? So the reason is because it have protein synthesis machinery. It have protein synthesis machineries. And who are in protein synthesis machineries? That is DNA, RNA, right? Enzymes for protein synthesis. Present. Getting the things or not? DNA, RNA, enzyme for protein synthesis are present, and that is the reason we are telling which one. And of course, how protein synthesis will occur without ribosomes. So you can say DNA, RNA, and of course ribosome. These all are present in which one? Mitochondria. And that is the reason we call it is a semi-autonomous organelle. Why it is not autonomous? Because it not have an independent existence. Some of the nuclear protein also regulate it. Got the idea? So it like you are working as a part-time job. You are not 100% dependent on your parent. You are earning somehow. Suppose that your expenditure is 20,000, 10,000 you are earning, 10,000 parent is helping. You are getting the idea or not? So it can synthesize some of the required protein, not all the proteins. And that is the reason we called it what? Semi-autonomous cell organelle. DNA, RNA, ribosome, enzymes for protein synthesis present. And ribosome present is which type? 70S type in mitochondria. Getting the idea, understanding the idea. Now if we talk about mitochondria, it is a double membrane bounded cell organelle. Double membrane bounded cell organelle. You can use a tricks, apply a tricks here. Whoever double membrane bounded in the cell have DNA. Got the idea? Double membrane bounded is what? Mitochondria. Double membrane bounded is what? Chloroplast. Double membrane bounded is what? Nucleus. So whoever the double membrane bounded is there, they have what? DNA. Clear? Right? So what I am writing, it is a double membrane bounded cell organelle. If we talk about shape, so It is variable, but mostly it is a sausage shape or cylindrical. Getting the things? If we talk about number is also variable. More mitochondria present in cell involve in more metabolic activity. More number of mitochondria is present in metabolically highly active cell, where need of energy is more. So you can say like number of mitochondria depend upon activity of the cell. Is it clear? Is it clear? It's all right. So skeletal muscles, if you talk about muscle cell have more number of mitochondria, right? Because it is working, it is requiring a lot of energy. Getting the idea or not? So shape is also variable, depend upon the workload. Able to understand? And it is which one? Sausage shape or cylindrical in shape. And if you talk about number, more number of mitochondria present in cells who are metabolically active. Is it clear in the mind? It is all right. It's done. Now we are moving ahead. Now we are going to deal about the structure of mitochondria. A structure of mitochondria. So if you talk about the structure of mitochondria, it is visible after staining. Mitochondria structure is visible after staining. Getting the idea? And how we stain it? With a stain that is vital dye, that is genus green B. It is a name of stain. It is a name of dye who is staining the bacteria. Getting the idea? Now we are moving ahead. Now we are going to draw the structure of the mitochondria. Have a look here. So I'm just drawing a rough sketch here. So you just think like, you just have a look here. Suppose that this is a mitochondria. It's somehow cylindrical structure I drawn. And if you talk about the mitochondria, it have two membrane. So outer membrane and inner membrane. If you talk about inner membrane is highly enfolded. Inner membrane of the mitochondria is highly enfolded. You just think like it is the enfolding of the inner membrane. So this is I am writing which one? Outer membrane, and this is which one? Inner membrane. Inner membrane. Write the outer membrane also full name, no problem. So write here outer membrane. Outer membrane. Right. So between outer and inner membrane there are certain space are there, and this is called what? Mitochondrial or even membranal space, or or you can also say perimembranal space, not a problem. Got the idea? So these are the space. If you talk about this inner membrane is highly enfolded and have another name, and another name is cristae. So inner membrane is also called cristae, and you can look, it is highly infolded. Is it clear? And if you talk about inner membrane, it have two type of phase. These are fluid filled matrix. These are what basically? Fluid filled matrix present in the which one? Mitochondria. This fluid is called what here? Basically matrix. So the face of the which one? Inner membrane toward the matrix is called M phase. And who are toward the cytoplasm is called C phase. Cytoplasmic phase. Got the idea? So two phases are there in the cristae, M phase or C phase. M phase is toward matrix and C phase is toward cytoplasm. Now, who is present in the inner membrane? So, at every distance of 100 angstrom, at every 100 angstrom distance, I'm not drawing everywhere, I'm just simply saying you, at every 100 angstrom distance, electron transport particle is present. Getting the thing? So these are what basically electron transport particle. We also called it what? Oxysome. So in these electron transport particle, getting the things or not? Here it is F0 and F1 particle is present. F0 and F1 particle is present. These are F1 and F0 and F1 particle. If you go for a detailed structure of it, I'm also giving you the idea. I'm taking this part of the membrane. Suppose that this is the inner membrane. This is the inner membrane. In inner membrane, what you are finding, these are who are embedded in inner membrane is called F0. F0. And who are protruded in the matrix is called which one? F1. We called it F1, or we also write CF1, coupling factor F1. And here it is an enzyme present. I am changing the color even for the enzyme. Have a look here. Enzyme is present here, and that is which one? ATP synthetase. Got the idea? So these are the detailings I have given for this electron transport particle or oxysome. And it is present at a distance of how much? Present at a distance of 100 angstrom. Of 100 angstrom. Got the idea? Understand the idea? So these are about the membrane, right? Both the membrane are made up of phospholipid bilayer. Both the electron membrane are made up of which one? Phospholipid bilayer. But both the membrane contain different enzymes. Both the membrane is made up of phospholipid bilayer like plasma membrane, but they contain different enzyme. Different enzyme. And I also want to write one thing here. Outer membrane of mitochondria, outer membrane is more permeable than inner membrane. Anybody can answer why? Outer membrane is more permeable than inner membrane. Anybody can write? Why is it? Very good answer. Very good answer. Porins are present in the outer membranes. You have studied in the transport of plant, na? Very good. Right. Impressive. So, Get the idea? It is written in outer membrane of mitochondria, plastid, and even in bacterial membrane. A particular proteins, porins are there, and which have higher, which enhance their permeability. Getting the idea? So that is about the membrane. After this membrane, what we are going to deal in the mitochondria? In mitochondria, who is there? Just have a look. In mitochondria, 70S ribosome is there. What happened? Why not selecting? So here who is there? 70S ribosome is there. So I'm just making here. It is 70S ribosome. 70S ribosomes are present. You better know DNA is present. Now if anybody asks a question, DNA is what? It is DNA present. It is a DNA of the mitochondria. So DNA is what? It is double-stranded and circular. And you can also say DNA is what basically? DNA is single, double-stranded and circular. Like the prokaryotic DNA. Getting the idea or not? Is it clear? So DNA is single, double-stranded and circular DNA is present here. Of course, I already wrote earlier, RNA, enzymes, protein synthesis enzyme, all things are there. Entire protein synthesis mechanism is there. Plasmid is not present here. Plasmid is not present here. One student asking, what about plasmid? So plasmid is not present here. So you got about the internal structure, what is present inside it. Now we are going to deal about function. Now we are going to deal about function of the mitochondria. Structure part is over. Now we are going to talk about function. So if you talk about the function of mitochondria, so you can say about cellular respiration. Cellular respiration. Now I am just telling you detailing. In cell respiration, how many components are there? Glycolysis is there. Wait, I am writing the component. Glycolysis, link reaction, Krebs cycle, and ETS, electron transport system, and oxidative phosphorylation. Oxidative phosphorylation. These are the steps of which one? Cellular respiration. This step occur in cytoplasm. Which step occur in cytoplasm? Glycolysis occur in cytoplasm. Remaining all step occur in mitochondria. So when burning of the food occur, major steps during burning of the food, cellular respiration occur in which one? Mitochondria, except glycolysis. Every steps occur in mitochondria. Link reaction and Krebs cycle occur in matrix of mitochondria. Right? Matrix of mitochondria. And if you talk about ETS on oxidative phosphorylation occur in which one? Inner membrane of mitochondria. Inner membrane of mitochondria. Got the idea? Is it clear in the mind? So in cell respiration, except glycolysis, everything during the burning of food occur in which one? Mitochondria. Like link reaction, Krebs cycle, ETS, oxidative phosphorylation, it occur in mitochondria. Out of that, link reaction and Krebs cycle occur in which what? Matrix. We will deal in very detail during which one? Cellular respiration. And ETS and oxidative phosphorylation occur in which one? Inner membrane of mitochondria. Is it clear in the mind? It's all right. And beside of that, I am just telling you another function. That is mitochondria is also associated with cytoplasmic streaming. Cytoplasmic inheritance, not streaming. Cytoplasmic inheritance. Cytoplasmic inheritance. two type of inheritance occur. Gene transfer occur from parent to progeny. When nucleus DNA transfer the character in next generation, it is called nuclear inheritance. And when DNA present in cytoplasm, like mitochondria, plastid, play a role in inheritance, it is called cytoplasmic inheritance, or it is also called extranuclear inheritance. Extranuclear inheritance. So it also have a role in these extranuclear inheritance. Able to understand? Clear in the mind? So this is about what? Mitochondria. So you got the function. Major part of the burning of food, cellular respiration, except glycolysis, occur in which one? Mitochondria. Out of that, link reaction and Krebs cycle occur in matrix. And obviously, common sense. Where the enzyme for link reaction and Krebs cycle present? Matrix. ETS occur in which one? Electron transport system or oxidative phosphorylation occur in inner membrane. So all the machinery associated with that present in which one? Inner membrane. Getting the things all complex we will study in which one? Cellular respiration chapter. Is it clear in the mind? It is all right. So who is over? Mitochondria is over. Right? Done. Now we are moving toward the another cell organelle, that is plastid. One thing I forget to write, that is how it divide, formed by which process? Binary fission. Formed by which one? Binary fission. Getting the things or not? Right? Clear? Means one mitochondria divide and two mitochondria form. Binary fission means what? This is a mitochondria, and from this mitochondria they are dividing and forming a which one? Another mitochondria. Got the idea? Clear about the right? So mitochondria over. About origin I am not telling, right? Because nowadays question not going to ask. If anybody going to ask, it have an endosymbiotic origin. Means in prokaryotic cells, some bacteria like iron sulfur bacteria have entered in prokaryotic cell, and later they permanent settled there and become mitochondria. Getting the things or not? Understanding the idea? So a prokaryotic cell got which one? Another prokaryotic cell, like you can say like, and later on during evolution of eukaryotic cells it occurs. Getting the things? Is it clear? Now we are moving ahead. Now I am coming on plastid. Have a look here. We are going to deal about plastid. If you talk about plastid, it is also a which one? Double membrane bounded structure, right? Double membrane bounded. Okay. If you talk about it is also a which one? Semi-autonomous. Autonomous cell organelle. Is it clear? Semi-autonomous. Here also who is present? DNA, RNA, ribosome, and of course here also which one? 70S ribosome, and other protein synthesis machineries are present. Other protein synthesis machineries are present. Got the idea? So like mitochondria, it is also a which one? Semi-autonomous. So how many semi-autonomous cell organelle are there? Cell organelle are there? Two. One is mitochondria, another is which one? Plastid. Is it clear in the mind? So now we are moving ahead. So if we talk about, we classify the plastid into three categories. Plastid into three categories. based on the pigments. Getting the things or not? Based on criteria is which one? Based on pigment. Pigment. So if we talk about plastid, it is leukoplast. It is chromoplast. And it is chloroplast. So we report three type of plastid: leukoplast, chromoplast, and chloroplast. In leukoplast, who is absent? Pigments are absent. Pigments are absent in leukoplast. It is colorless plastid. Colorless plastid. Getting the idea or not? It is a colorless plastid. If you talk about the leukoplast, function is what? Storage. Function of leukoplast is storage. Got the idea? Understand the idea? Leukoplast further, it is of three type. Based on what it is storing. It is amyloplast. It is aleuroplast. It is also called proteinoplast. And another is elioplast. So based on what it is storing, these leukoplast is of three type. Getting the things or not? If you talk about the amyloplast, it store starch, or you can say carbohydrate. Starch is a which one? Carbohydrate. In bracket you can write carbohydrate. So amyloplast. Aleuroplast store protein. It store protein. And if you talk about elioplast, it store oils and lipid. Got the idea? So these are about the first type of plastid, that is what? Leukoplast. Is it clear? Right? Now we are going to talk about which one? Chromoplast. It is colored plastid, but other than green. If you talk about chromoplast, it is colored, but color is other than what? Green. So colored plastid. Getting the things. Who is present? What are the pigment present in the abundant amount, more amounts in case of chromoplast? So carotene and xanthophyll mainly. Got the idea or not? Carotene and xanthophylls are mainly present in which one? Chromoplast. So you can say about the chromoplast, example where it is present. You find a tomato, red color. Getting the things is due to which one? Chromoplast. Chili color is due to which one? So in tomato, who is there? Which pigment is there? Lycopene. Lycopene is there. Getting the things or not? Who is there? Lycopene is there. Petals have different colors, na? Petals of flowers have different color. So another example you can say petals of flower have variety of coloration. Able to understand? These are dominating in which one? Carotene and xanthophyll. Now we are coming on which one? The third one, and that one we will deal in detail, and that is which one? Chloroplast. If you talk about, it is green color plastid. Green color plastid. And who is the pigments present in chloroplast? So pigments present in the chloroplast is chlorophyll A, chlorophyll B, carotene xanthophyll. But it is green because of chlorophyll A and B present in which one? More amount. In compared to carotene and xanthophyll. Able to understand or not? Clear in the mind? So that is the reason it is green color plast. Is it clear? Now we are going to deal about the structure of chloroplast. So I am asking a small question. Try to answer it. Tell me a statement is true or false. So chloroplast is present in all plant cell. Is it correct or wrong? It is a false statement, not a true statement. Chloroplast not present in all plant cell. Chloroplast present only in which plant cell? Green plant cell. Green part of plant. So if you talk about chloroplast present in, where it is present? Present in green part of plant. The part of the plant which is not green, green part of plant. Able to understand? So part of the plant who is not green, chloroplast is not there. Is it clear? Let me tell you something. Wait a minute. I am writing here proplastid. Proplastid is precursor of plastid. So every type of plastid form from which one? Proplastid. Getting the idea? So proplastid is precursor of plastid. New plastid form from which one? Proplastid. And now I am writing here, suppose that here chloroplast is there. Here leukoplast is there. Here leukoplast is there. And here chromoplast is there. So see, leukoplast may convert into chloroplast, chloroplast may convert into leukoplast, leukoplast may convert into which one? Chromoplast, and chloroplast may convert into which one? Chromoplast. Able to understand or not? Is it clear? So leukoplast convert into chloroplast, chloroplast convert into leukoplast, chloroplast also convert into chromoplast, leukoplast also convert into chromoplast. But once chromoplast form, it not convert into chloroplast and leukoplast. Getting the idea? Understanding the idea? Light. So if leukoplast getting light, in presence of light, it convert into chloroplast. Chloroplast for long time not getting the light. It convert into which one? Leukoplast. Clear? Understanding the idea? So they are interconvertible. Once condition is what? Once the chromoplast not formed, further it remain chromoplast. Chromo to chloro it not become. Once tomato become red color, it not once become green color. Na? But green color tomato convert into which one? Red color tomato at the time of ripening. It happen or not? Is it clear? It is all right? Yes. So now we are going to deal about the structure of chloroplast. Structure of chloroplast. Give your attention. So if we talk about the structure of chloroplast, it is also a double membrane bounded structure. So this is two membranes of the chloroplast. Okay, this is outer membrane. And short I am writing inner membrane. And inside the inner membrane, who is there? Fluid filled structure is there, matrix is there. But here this matrix is called what? Stroma. Matrix, fluid part is called stroma. And inside that, what you are finding, who is also present? DNA is present. Double-stranded DNA, DS, circular DNA. Got the idea? Ribosome is present. Which kind of ribosome? We can say 70S type of ribosome is there. 70S ribosome. Now I am telling you very important things here. Inside the chloroplast, you will find there is a stack-like arrangement occur of membranous network. You find like many coin-like structures are there, who are present, piled up, present one above the others. Got the idea? Understanding the idea? So these are called granum. One piles is called granum. And because many granums is there, so plural of the granum is what? Grana. Getting the idea? So here plural is what? Grana. It is singular and it is plural is which one? Grana. Is it clear or not? So granum, grana, one piles of coin-like a structure, membranous enfolding. One is called granum. More than one are there. It is altogether grana. It is singular and plural. Now these granums are joined together with another membranous structure like this, and this is called stroma lamella. A stroma lamella. And now granum, unit of granum, is thylakoid. Who is there? Thylakoid is there. So if you talk about this one is a thylakoid. I'm just drawing outside. So this one here this is a thylakoid. Are you getting the idea or not? This is a thylakoid. And many such kind of coin-like structure, circular membranous structure piled up over each other and forming a granum. So this is a thylakoid. Let me write. This structure is a thylakoid. Thylakoid. So now question arises, where the photosynthetic pigments are present in chloroplast? Where the photosynthetic pigments are there? So you can write photosynthetic pigments are present in thylakoid membrane. Thylakoid membrane. Got the idea? Thylakoid membrane containing photosynthetic pigment. And this thylakoid membrane is also called frit membrane. This thylakoid membrane is also called which membrane? Frit membrane. Is it clear in the mind? So here these photosynthetic pigments are present as photosystem. In photosynthesis I will tell you. So these photosynthetic pigments are present as which one? Photosystem, like PS2, PS1, getting the things or not? So these are the pigments present in the thylakoid membrane. Now what I am telling, here he is what basically? Lumen of thylakoid membrane. Lumen of thylakoid membrane. Outside of this, who is there? A stroma is there. If you talk about outside, who is there? Stroma is there. So many thylakoid membrane, you just I am drawing up one more diagram. Many thylakoid membrane pile up like this and they are forming which one? Granum. One above the other and they are forming which one? Granum. Are you getting the idea, understanding the idea or not? So now if we talk about photosynthesis, if we talk about photosynthesis, photosynthesis have two components. You better know. One is which one? Light reaction. Another is which one? Dark reaction. Dark reaction. So the light reaction of the photosynthesis, where it occur? Occur in grana. Why occur in grana? Because pigments are there. Pigments are there. Getting the things or not? To attract whom? Sorry, to absorb whom? To absorb. Pigments are there to absorb which one? Sunlight. And dark reaction occur in stroma. Dark reaction occur in stroma. Getting the idea, understanding the idea, how these light reaction occur, how these dark reaction occur, entire detailed mechanism you will study in which one? Photosynthesis chapter. We will talk in very detail how this light reaction occur. So simply here you keep in mind light reaction occur in grana because in grana photosynthetic pigments are there. Dark reaction occur in stroma. Enzymes for dark reaction also present in which one? Stroma. Enzymes for dark reaction present in stroma. Occur in reaction or present in stroma. Like mitochondria, so it is one of the function. Another function you write, cytoplasmic inheritance. It also have a role in which one? Cytoplasmic inheritance. And I already told you how the new plastid form. New plastid form from which one? Proplastid. New plastid form from proplastid. Is it clear? It is all right? So who is over? Chloroplast also over, plastid also over. Now we are going to discuss about ribosome. Okay, ji? Ribosome. Give your attention. Ribosome. If we talk about ribosome, it is also called pallid particle because it is discovered by whom? Pallid. Or it is also called cell organelle within a organelle. Because of it is the only cell organelle present in another cell organelle. So if you talk about ribosome, it is a non-membranous structure. Non-membranous structure. And if you talk about ribosome, is made up of two things. Ribosome is made up of two things together. Ribosomal RNA plus protein. So rRNA plus protein together forming which one? Ribosome. Got the idea? So ribosome is made up of ribosomal RNA and protein. Clear? Is it all right? It is done. Now I am telling type of ribosomes. So if you talk about the type of ribosome, it is of two types. One is 70S type of ribosome. Another is 80S type of ribosome. So if you talk about 70S ribosome is made up of two subunits. The smaller subunit here is 30S and here larger subunit is 50S. Don't use mathematics, sir. 50 plus 30 equal to 80. S means Swedberg unit. Here S stand for Swedberg unit. And you can say it is the unit of sedimentation coefficient. Sedimentation coefficient. And you can say indirectly it is measuring the density. Who is more denser will settle faster. sediment faster. So you can say density and size. It is indirectly saying about density and size. So who have less density, 30s, who have more density, 50s, getting the idea. Now I am just telling you 30s, 50s, and if you talk about 30s, so 30s have what basically? Which type of ribosomal RNA? 16S rRNA is present in 30S. 16S rRNA is present in 30S. Getting the things. And in 50S, the type of RNA present in, rRNA present in, which one? Two type. One is 23S, another is 5S. So two type of ribosomal RNA present in 50S. Now I am going to talk about 80S. It is also made up of two type of subunit, 40S and 60S. If you talk about 40S, 18S type of rRNA is there. Which ribosomal RNA is there? 18S. And if you talk about 60S, have three type of ribosomal RNA. One is which one? 28S. One is 5.8S. And another is which one? 5S. Another is 5S. So these are three type of ribosomal RNA present in which one? Getting the things. If we talk about 70S ribosome, in 70S ribosome, 60% rRNA is present, plus 40% protein is present. If you talk about 80S, here 40% rRNA is there and 60% proteins are there. Got the idea? And now which rRNA present here? Getting the things. Now I am just telling you, here 23S is a ribosome and form peptide bond during protein synthesis. When you will study second year, we will talk about peptide bond formation. So these are 23S act is what basically? Peptidyl transferase. So it is peptidyl transferase enzyme in prokaryote. And in eukaryote, this is peptidyl transferase. 28S is peptidyl transferase. Getting the idea or not? So peptidyl transferase in eukaryote is 28S and in prokaryote it is 23S. About this in very detail we will study during which one? Protein synthesis in second year topic. Getting the idea? Simply I have given a basic idea. Now two ribosomal subunit remain separate or they join together. Getting the things, assemble together. This depend upon the concentration of a cofactor. And what is this? Which ion is there? Magnesium ion. Getting the things or not? Magnesium ion concentration will decide ki either who two ribosomal subunit will separate or it remain attached together. And even magnesium ion concentration, if it is high concentration, getting the things or not, then in that case what we are finding, two ribosomes join together and forming a dimer. Forming a which one? Dimer of ribosome. Dimer of ribosome. So these magnesium ion concentration decide that the two ribosomal subunit will assemble or deassemble or if it is in higher concentration, what happened? Two ribosomal subunit, two ribosome to join and forming a which one? Dimer. Getting the idea or not? Now what are the function of ribosome? Function of ribosome is protein synthesis. Protein synthesis. Got the idea? So it is a cell organelle associated with protein synthesis. Is it clear? Now we are going to talk about cytoskeleton. Cytoskeleton. Can we proceed? A lot of things are there, na? So that is the reason if we not maintain the speed, things will not finish. Right? So move. So now we are going to talk about cytoskeleton. Cytoskeleton. If we talk about cytoskeleton, NCERT wrote about the three things. It is a proteinaceous structure. First you think proteinaceous structure. Got the idea? And three type of cytoskeletal structure are present in the cell. So the first I'm writing microtubule. Microtubules. Another I'm writing microfilament. Filament. And third I'm writing intermediate filament. And NCERT wrote also these cytoskeleton have a role in which one? Support shape of the cell, holding the cell organelle, locomotion, cell division. Getting the idea? So these are the variety of job done by which one? These cytoskeleton. Able to understand? Of course, motility, locomotion, support, shape, holding the cell organelle, locomotion, cell division. So these are the some of the area where these cytoskeleton play a role, have a role. Getting the things or not? Now, if we talk about these cytoskeleton, so I am telling first you about the microtubule. So if you talk about microtubule, it is a tubular structure. And tube means hollow, है ना? Tube means hollow. So it is a hollow tubular structure. It is a hollow tubular structure. You just see. This is the microtubule. Diameter is how much? 25 nanometer. Its diameter is 25 nanometer. Understanding the idea or not? It is a tubular structure. Microfilament is a filamentous structure. Filamentous structure. So microfilament, you will find it is like this. So it is not a tubular structure, it is a filamentous structure. Getting the things or not? An intermediate filament, it is fibrillar structure. Fibrillar structure. Intermediate filament are joined by like this. N terminal of protein, C terminal, N terminal, 2 C terminal, N terminal, then further NC, then CN. Getting the thing? So it joined like this. Fibrillar structure, joined like this. You have seen the coconut coir. Coconut coir. So how the coconut coir is arranged, same manner intermediate filaments are arranged. Getting the things? So if we talk about microtubule, microtubule made up of tubulin protein. Made up of tubulin protein. Tubulin protein. Microtubule is made up of which protein? Tubulin protein. Microfilament, it is made up of actin protein. Made up of actin protein. Actin protein. Getting the things or not? An intermediate filament made up of keratin protein. Keratin protein. Who is made up of keratin protein? Intermediate filament made up of keratin protein. So it is how much nanometer? I told microtubule it is 25 nanometer. And if you talk about which one? Filamentous structure, it is how much? 7 nanometer approx in the thickness. And here it is 12 to 15 nanometer. 15 nanometer. So why we are telling it intermediate? You got the idea? Why I am telling in which one? Intermediate filament as intermediate, you got the idea? In their diameter, its diameter is in between microtubule and microfilament. Who is the smallest? Size is intermediate, of course, very good. Its size is intermediate. So I wrote the size 12 to 15 nanometer, 25 nanometer and 7 nanometer. Getting the idea, understanding the idea. So these are about. Now if I talk about microtubule, made up of which protein? Tubulin protein. Microtubule is made up of which one? Tubulin protein. So if you talk about tubulin protein, if you talk about tubulin protein is made up of two type of protein. Alpha tubulin and beta tubulin. Alpha tubulin and beta tubulin. Are you getting the idea? Understanding the idea? Alpha and beta tubulin. And both alpha and beta tubulin alternately arranged in a protofilament. Who alternately arrange? Alpha and beta tubulin arrange alternately in which one? Protofilament. Now I am telling you a structure of microtubule, then you will understand it better. See, I am just making a tube here. Look here. This is a tube. This is a tube. Agree? This is a tube. Here it is a tube. So this tube-like structure is made up of which one? Protofilament. And how many protofilaments are there? Here the number of protofilament is vary. In some microtubule the number of protofilament is 13. In some microtubule the number of protofilament is 10. Look here. I am drawing here one protofilament, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13 protofilament. It is 13 protofilament. You just try to understand. Getting the things. So if you talk about, I am just drawing here, have a look here. I am once again drawing a diagram. I am writing 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, space is less, 12, 13. An example. I am drawing here, it is alpha tubule. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13. It is the, so it is, you just try to understand. You just try to understand here, one important things I am telling you. This is the one protofilament. Wait a minute, make me draw a line. This is one protofilament, it is second protofilament, it is third protofilament, it is fourth protofilament, and likewise, everywhere, alpha, beta. So can you say this is a one protofilament? It is a protofilament. So who is present? If you talk about alternate arrangement, I told na, then once who come? Alpha. You just think like, then also who come? Beta. You just try to understand. Then who come? Beta. You are getting the idea. So who is alpha, beta, alpha, beta? So I wrote one line, you got the idea or not? This protofilament is made up of alternate arrangement of alpha and beta tubulin. So in each protofilament, one protofilament, likewise 13 protofilament. So you see now, it is one protofilament. Two color I am telling. It is second protofilament. It is third protofilament. Likewise, 13 protofilament. And you just cover it, wrap it. So first it become a sheet. When you wrap the sheet, it become a tube. It become a tube. Are you getting the idea or not? So first it is forming a sheet-like a structure, then it is converting into a tube-like a structure. So you got the idea. You understand about these microtubular structure, understanding the idea, clarity came or not. So simply line of NCERT can't help you there. Simply NCERT wrote microtubule, microfilament, intermediate filament are what basically making the cytoskeleton. Now entire dissection of microtubule came or not. It is made up of tubulin protein, two type of tubulin protein, alpha and beta tubulin protein. So picture of the microtubule came in the mind. Now we are coming on cilia and flagella. Okay, ji, can we start? Look here. So we are going to deal about cilia and flagella. So cilia and flagella, if you took, they have different external structure. External structure, but same internal structure. So first I am telling you what are the difference in the cilia and flagella externally. So I am writing here cilia and here I am writing flagella, and I am writing some differences first between cilia and flagella. So you look the differences between cilia and flagella first. Have a look here. So if you talk about cilia, it is small. Flagella, it is large. Long, you can say. Long. So one, cilia number if we talk, many in number. Number is also different, size is also different. So number many. Here if you talk about flagella, one to few. If you talk about location, present all over body surface. Body surface. So mostly anterior in position. Mostly anterior in position. So now if we talk about some more difference, beating of cilia, two type of beating is there, synchronous and metachronous. Synchronous and metachronous beating. Beating of flagella is independent. If you talk about movement of cilia, we called rowing movement. Rowing movement present in cilia, in flagella undulatory movement. Undulatory movement is present in which one? Flagella. Rowing movement of two type of a stroke, power stroke and a recovery stroke. So like a swimmer, when you know swimming, how to swim. So when you swimming in water, you are putting your hand in water like this. So it is which stroke? And pull the water. It is power stroke. So when you go for power stroke, you move forward. and then you different way you just remove your hand. It is recovery stroke. So it is power stroke, and then you are coming once again, it is recovery stroke. Getting the idea? Understand? Is it clear in the things? Is it all right? So undulatory movement. Undulatory movement, here it create a waves. It create, when you jerk a whip, what happen? A waves create. So it is called which one? Undulatory movement. Here movement is power stroke and recovery strokes. Now if we talk about cilia types. So two type of cilia we are just finding, kinocilia and stereocilia. If you talk about kinocilia, have low role in movement, and stereocilia act as a receptor. So kinocilia, where it is present? It is present in trachea. Kinocilia, where it is present? It is present in fallopian tubule and moving the which one? Egg. In trachea, it is expelling the dust particle. Are you getting the idea? And stereocilia, it act as, it is in your tongue. This is what? Stereocilia. So it is present in the taste bud. Gastratoreceptor. Getting the things or not? So taste bud. Gastratoreceptor have which one? Gastratoreceptor have which kind of cilia? Stereocilia. Got the idea? Now I am talking about flagella. So flagella I am also writing two type. One I am writing whiplash flagella. Another is tinsel flagella. So if you talk about whiplash flagella, it like a whip. Base is broader and apex is narrower. And tinsel flagella have everywhere same diameter. And here bristles are there. So these are the some of the difference between cilia and flagella I told you. You got the differences between cilia and flagella. Is it clear in the mind? Okay, ji, done. Now I am going to say about the similarity between cilia and flagella. Similarity. And similar is what? Their internal structure. Similar is the internal structure. Got the idea? Understand? Doubt I will give you my number, you just ask, because if I'm just going to work on that, all time will over. 9680274846. This is my mobile number. You can put the WhatsApp, right? I will just answer it. Okay, ji? So whiplash and tinsel I told na, same diameter. Here broader base and narrower apex. Here everywhere diameter is same and bristles is there. So it is tinsel, it is whiplash. Getting the things clear? Now I am moving ahead. Have a look here. Now I am going to move ahead. Just give your attention. See the things. Ultrastructure of cilia and flagella. Their internal structure we are going to this. So one is what? Periplasmic membrane. Two component is there, periplasmic membrane, and another component present in the cilia and flagella is axoneme. Or we also called it what? Axial filament. So this periplasmic membrane is what it is? It is plasma membrane. Covering the cilia and flagella. Covering the cilia and flagella is called periplasmic membrane. You just look now, this is the cell membrane and this is the basal body and this is what? Cilia or flagella. So what you will find, here plasma membrane extended like this and it is covering some part of the cilia and flagella, and this is called periplasmic membrane. So axoneme and axial filament, getting the things or not? So these axoneme and axial filament have which one? 9+2 arrangement. These have which arrangement? 9+2 arrangement. So 9 peripheral doublet is there. 9 peripheral doublet is there. Getting the things or not? And these two doublet, it is what basically? Microtubule. 9 peripheral. I told about microtubule. Now 2 central microtubule. Two central microtubule. So 9 plus 2 arrangement is there. So if you talk about peripheral doublet, you look here, I am drawing a diagram. This is a one microtubule, another microtubule attached like this. Another microtubule with the first microtubule arranged like this. So these peripheral doublet, it is a doublet. So it is a doublet. If you talk about, it is made up of 13 protofilament. And it is made up of 10 protofilament. How many? 10 protofilament. So it is called A tubule, it is called B tubule. And from A, who is there? Arms are arising. Arms is arising. Arms is attached with which one? So which one? A tubule. So this is peripheral doublet detailing. Peripheral doublet. Peripheral doublet is made up of A and B tubules. A is made up of how many? 13 protofilament. B is made up of which one? 10 protofilament. Getting the idea? Is it clear? 13 and 10 protofilament. Is it clear? And arms is attached with which one? A filament. Got the idea? Is it all right? Now I am going to tell you about the structure, ultra structure of the which one? Cilia and flagella. Give your attention. Ultra structure. Have a look here. So here it is 2 central microtubule. This is called C1, this is called C2. C1 you will find some hair-like granular depositions, a structure arising. In C2 it is not there. So you can differentiate C1 and C2 like this. And this is the cross bridge. This is the cross bridge between C1 and C2. You can say it is C1, C2 linker. So these are the 2 central microtubule, and these 2 central microtubule is surrounded by which one? Central sheath. So this is central sheath. It is central sheath. It is surrounding the which one? It is surrounding the C1 and C2. Now how many doublet are there? Now how many doublet are there? 9 doublet are there. 9 peripheral doublet are there. So let me make a imaginary circle. Otherwise it not become circular. Let me just a minute. Give me a minute. Okay, ji. So now see, I am telling 9 peripheral doublet. So this is 1 doublet, 2nd doublet, 3rd doublet, 4th doublet, 5th doublet, 6th doublet, 7th doublet, 8th doublet, and this is 9 doublet. And this blue color is nothing. It is I draw a line to make you understand कि भाई line becomes somehow circular. Got the idea? So 9 doublet are there. These are how many doublet? 9 doublet are there. 9 doublet is there. Got the idea? Now these doublets, inner side, somehow inner side A is there, somehow outer side B is there. A tubule, B tubule. Everywhere I will not write. So see, A and B is going to join together. A and B are joining. Who is joining? A and B are joining. Inner side you think it is A, it is B. So here A and B is joining. 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 So A and B is joining. Getting the thing? So it is called AB linker. It is called what? AB linker. Are you getting the idea? Understanding the idea? It is AB linker. An entire structure together called exoneme. Wait a minute. Now from A, how many radial spokes arise? 9 radial spokes are arising from the A like this. From A microtubule, who is arising? Radial spokes are arising. From A, radial spokes are arising. So how many total radial spokes are arising? 9 radial spokes are arising. And from A, arms is also attached. Na? I already told na, arms are attached with which one? A. So A, outer arm, inner arm, inner arm, outer to outside of AB linker, and inner inside of the AB. Outer arm, inner arm, outer arm, you can say like inner, outer, inner, outer, inner, outer. So these are the arms which is arising. Are you getting the idea? So this is the structure of a exoneme, 9 plus 2 arrangement. Now I am just labeling and I am just writing protoplasmic sheath. So around this, this is a plasma membrane and which one? I wrote na plasma membrane and periplasmic membrane. It is periplasmic membrane or plasma membrane. Periplasmic membrane and plasma membrane. Now I am writing the name of everyone. It is what? It is doublet. Doublet. It is arm. Outer side, outer arm. Inner side, inner arm. This is AB linker, I already wrote. AB linker. This is central sheath. Central sheath. These are which one? Cross bridge. Cross bridge. And these two are central singlet. Central singlet. These two are central microtubule. So central singlet. C1, C2, both are central singlet. Able to understand? These are radial spoke. These are radial spoke. Got the idea? Understand the idea? Now, I am telling you the composition, chemical nature. It is very important. So, doublet is made up of microtubule. Central singlet doublet is made up of which one? Microtubule. Now, these arms is made up of dynein protein. Arms is made up of dynein protein. AB linker is made up of nexine protein. And these radial spokes is also made up of protein. But chemical nature is not defined, name is not defined which proteins. Able to understand? Getting the idea? So this is about which one? Cilia and flagella. And you keep in mind, these cilia and flagella arise from centriole-like structure called basal bodies. So cilia and flagella arise from which one? Centriole-like structure called basal bodies. Get the idea? So this is about cilia and flagella. Now centriole is missing. What to do? Want to do today and next class. Because today it is up to centriole. See, even I move faster. Not able to reach, na? Because it is only possible to say when I say the NCERT line only. Otherwise, diagram, drawing, and about the which one, microtubule, microfilament, of course, that clarity required. Na? So everything came in your mind. Everything came in your mind about cilia, flagella. Any confusion is there? It's all right. So you draw it along with getting the thing. Then the perfection came. Draw the diagram, label the diagram. So nexine protein. Who is made up of nexine protein? Arms. Who is made up of dynein protein? Sorry, dynein protein made up of arms is made up of dynein protein, and arms is made up of dynein protein, and AV linker made up of nexine protein, and outer peripheral doublet is made up of which one? Tubulin protein. So how many type of proteins are present in cilia and flagella? 3 types, na? Got the idea? Dynein also present, nexine also present, tubulin also present. Now, but in NCERT wrote only tubulin protein. So if any question is coming which is very superficial, can see cilia and flagella, which protein is there? Tubulin, dynein, nexine, and all of these are not in option. Then what you will choose? Then what you will choose? What is it? Right, tubulin you have to choose. If it is all and these is given, you have to choose which one? All of these. Getting the idea? Understanding the idea? So I hope you understand about the cilia and flagella. Okay, ji. Bye-bye. Have a good day to all of you. QR code.